Hello, viewers. Welcome to Biodrunks. Let's learn some aspects of biotechnology. For your reference, I'll be providing notes, PDF link in the description. This video is also available in Hindi and Telugu. For more videos and continuous learning, please like, share, and subscribe. Let's start. In today's video, we are going to continue with tools used in gene cloning. We already dealt with the topics like nuclease, DNA ligase, and polymerase, and today we are going to do DNA modifying enzymes. So why are DNA modifying enzymes very useful? See, whenever you have a particular process in gene cloning, so practically things doesn't go the way it looks theoretically. So for the things to go practically the same, we need some modifying enzymes. For example, the DNA should not self-ligate. The vector should not self-ligate. We need this as a recombinant molecule. Okay, but sometimes what happens is it gets self-ligated. Okay, like this. It gets self-ligated. So we are not getting a recombinant molecule. Okay, and we don't need that process to be going on there. Or we want to check if it is properly ligated to the main source or not. Okay, we need to even check that. Another thing we need to check is, okay, they got ligated to the proper thing, but what if the polynucleotide or the phosphate ion is not there present properly? Okay, so we need to check all of these. And the reason we are using tools in gene cloning, that is a DNA modifying enzymes. Let's see what are the different DNA modifying enzymes. So different DNA modifying enzymes are alkaline phosphatase, polynucleotide kinase, and terminal transferase. Let's learn one by one in detail. Alkaline phosphatase. So alkaline phosphatase is a hydrolyzed enzyme which is responsible for removing phosphate groups, okay, from many types of molecules. Now the process of removing phosphate group is called as dephosphorylation. So in short, alkaline phosphatase works on the principle of dephosphorylation. Okay, and it is called as alkaline phosphate because it works at an alkaline pH. It works at an alkaline pH and removes phosphate ions. Okay, now where are these alkaline phosphates obtained from? They are obtained from bacterial alkaline phosphate, or we can call it as BAP. Okay, so it's the most active of the enzymes, but it is also very difficult to destroy. It's very difficult to destroy. Okay, and the reason is it's very difficult to destroy because it is highly active. It is mostly active. Okay, then we have calf intestinal alkaline phosphatase, or we can call it as CIP. It is obtained from the bovine intestine. Okay, it's obtained from bovine intestine, and it's not as active as our BAP. Okay, and this is mostly used in molecular labs. It's widely used in molecular labs because it can easily degrade once we are done with the reaction. Now you have shrimp alkaline phosphate, which is also called as SAP. Okay, what does this do? It is derived from cold water shrimp, and it can be readily destroyed by heat. That is, if you're using it for keeping it for 65 degrees Celsius for 15 minutes, it can be destroyed or degraded. Okay, so we learned about what is an alkaline phosphatase, what's its name, and where it is obtained from. Now let's see why it is used and where are we using it. See, as I told earlier, it removes phosphates. Okay, so removing phosphate from the plasmid and bacteriophage vectors, so that have been cut with a restriction enzyme. Now there is a bacteriophage vector, it's been cut with a restriction enzyme. Okay, so for the next ligation reactions, it does not allow it to self-ligate again. Okay, if you're removing it, so it won't self-ligate. Okay, otherwise there are chances that it gets ligated by itself. Okay, and Another reason why we use is because it removes from the 5' phosphate so that we can label it with radioactive phosphate. So what happens is the polynucleotide kinase, okay, which we are going to learn ahead, is used to do something called phosphorylating. Okay, now if you remove the phosphate, then only you can add a radioactive phosphate to it. That's the reason we are using alkaline phosphatase here. Next, we are going with polynucleotide kinase. So what does this do? As we were dealing with it, it is used to catalyze the transfer of terminal phosphate of ATP. Okay, terminal phosphate group of ATP to 5-hydroxyl terminal of polynucleotides. Okay, see, in the presence of ADP, it will also catalyze the exchange of 5' terminal phosphate groups and ATP. Okay, so this is commonly used to label RNA and DNA with 32P or 33P. So it is what it does, labels RNA and DNA with 32P or 33P at ends. Okay, and always remember, PNK is inhibited by ammonium ions. So ammonium acetate should not be used during the nucleic acid precipitation prior to phosphorylation. Okay. Now we know what does a polynucleotide do. Let's learn more about it. Polynucleotide kinase. Okay. Now see, the enzymatic activity of polynucleotide kinase is utilized in two types of reaction. One, we call it as forward reaction. Let's learn about forward reaction first. Okay, what happens in a forward reaction? Now this is our 5' OH 3'. Okay, now the PNK, I'm writing it as PNK, polynucleotide kinase. It transfers the gamma phosphate from the ATP to the 5' polynucleotide of DNA or RNA. So as you can see here, it has taken it. Now it is transferred it to 5' phosphate. Okay, as you can see here, this is the phosphate which has been removed from here. Okay, so this is called forward reaction. There is one more reaction called as reverse reaction or exchange reaction. We call it as exchange reaction mostly. So what happens in an exchange reaction is the target DNA or RNA that has the 5' phosphate is incubated with excess of ADP. Okay, now you have this as 5' phosphate and it has been incubated with excess of ADP. And in the presence of P and K, what happens? The P and K will first transfer the phosphate from the nucleic acid onto an ADP and forming an ATP. Okay, so now this has been done. So as you can see here, it has transferred it. Now this P, now this again, this has been done. Okay, so here you have ATP and it leaves behind dephosphorylated target. As you can see, it is dephosphorylated now. Now PNK then will perform a forward reaction and will transfer a phosphate from this ATP. Okay, again it will be PNK from this ATP to 5' phosphate. Okay, this is called as an exchange reaction. So you might have understood what is a forward reaction and an exchange reaction of P and K. Yes. So where are they used? Now let's know where is the P and K used. As I told you earlier, it's used in 5' labeling because I want to put a phosphate there. So I'm using it. Okay. Now, again, phosphorylation, because phosphorylation is addition of phosphate. Dephosphorylation is removal of phosphate. So here, phosphorylation. Here I can write it as addition of phosphate. Okay, at the 5' terminal prior to ligation. Now I want it to ligate, so I want the phosphate, otherwise it won't ligate. Now I want to know where is the terminal of RNA transcript. So marking the terminal and removal of 3' phosphodiol groups. Okay, so hope this is clear till here. Now we have the next one, which is terminal transferase. What is this terminal transferase now? So we call it as DNA nucleotidal transferase, terminal transferase, and it catalyzes the addition of nucleotides to the 3' terminus of the DNA. What does it do? It adds nucleotides, catalyzes, okay, adds nucleotides to 3' terminus of DNA. Okay, interestingly, it works on single-stranded DNA. Works on single-stranded DNA. Okay, including which has 3' overhangs or double-stranded DNA. That and that is an example of a DNA polymerase that does not require template. Does not require a template or primer. So it can add homopolymers or ribonucleotides to the 3' end of the DNA. Okay. Now the preferred substrate for this enzyme is 3' end, protruding 3' end. I can write it as protruding 3' ends. But it can also add nucleotides to blunt and 3' resist ends. And cofactor, which is very important for the reaction to carry out, is cobalt. Now, the enzyme can also catalyze upon magnesium and manganese administration. Okay. So till here we discussed about it. Now, what does this do? Let's find out what's the next thing. See, for example, this is my DNA. Okay, here it is 5', this is 3', this is 5', this is 3'. Now I am adding terminal transferase or terminal deoxynucleotidal transferase, or you can call it as terminal transferase. Okay, see, it is a template independent DNA polymerase, as it can synthesize DNA polynucleotide without the base pairing. Okay, now this enzyme is used for the formation of cohesive end by polymer tailing. Okay, homopolymer tailing. So homopolymer tailing is very important. Homopolymer tailing. So homopolymer tailing means a polymer in which the subunits are same. It has same subunits. Okay, this is called homopolymer tailing. Now, this transferases the catalyzes the addition of series of nucleotides onto the 3' OH terminal. So as I said, it will do at the 3' OH terminal. Okay, so as you can see, this is the DNA. How it will look now. Let's see. Let me take a different color for this. Here. For example, if it's adding C, 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 C. This is a 3' terminal. Here, this is a 3' terminal. Okay, C, 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 C. Okay. So, if this reaction is carried out in the presence of just one type of deoxynucleotide, a homopolymer tail is produced. Understood till here? Now, what are the applications? The applications of application of the terminal deoxynucleotidal transferase is it adds homopolymeric tails to the 3' end. We have seen it just now, and it labels the 3' end because as it is adding it the 3' end, so we are using it to label the 3' end of the DNA. So we have already dealt with all the important gene cloning tools. Okay, if any other tool is left, You can comment it out. This was all about today's video. Hope you liked it. For more such interesting video, please like, share and subscribe to Wired Rank. Let's meet in the next video. Bye-bye.